Hello, in this video, we're going to be talking about the concept of gene regulation, which is essentially controlling how much protein, a specific protein, is being made. So, for example, if a cell wants to make more of a specific protein or less of a specific protein, it can either slow down transcription or increase transcription, or can control translation. And both bacteria and eukaryotic cells control their gene regulation. So again, they're controlling how much of a specific protein is being made. And obviously, this is going to be based on their needs. So in a specific situation, a bacteria might need a protein, but in other situations, it might not. And so there's ways to turn these genes on and off so that you can have more or less of a particular protein. So one of the most efficient ways to regulate a gene is by regulating transcription because you are stopping it or increasing it from the beginning. Remember that we go from DNA to RNA first, that's transcription, and then RNA to protein, that's translation. But if we stop it or increase it at transcription, that's usually the better route. And so there's different ways to regulate transcription. So the normal way that a gene is used to make protein, well, in this case, to make RNA, is the RNA polymerase is going to bind to the promoter, which is at the beginning of the gene, and then it's going to make the mRNA out of it. And so in cells, they can either increase transcription or decrease transcription, which would effectively increase or decrease how much protein is being made. And so there are molecules called repressors that bind to the front of the promoter. It's actually right in between where the promoter and the gene is. And these repressors, because they're physically bound to the DNA, the RNA polymerase cannot produce mRNA. And so it either completely stops or at least slows down the rate of transcription for that particular gene. And then the opposite of that would be an activator. So activators typically bind to the promoter itself, which again is found on the DNA in the beginning of the gene. And these activators enhance or increase the amount of RNA polymerase that binds to that area, thus increasing transcription. And so this is just a straightforward way of either stopping / slowing down the production of a specific protein or increasing. And again, we are doing it from the beginning. So we're either increasing or decreasing transcription, which would effectively increase or decrease the production of that protein down the line. And with bacteria, they're much more efficient at doing this because they keep their genes in something called an operon. So operons are groups of genes that are regulated by the same promoter. And so here you can see there's gene A, B, C, D, and they are all regulated by the same promoter. So basically, they all have the same on-off switch. And so if A is on, then B and C and D are also all on at the same time. Now, this is going to be really useful. For example, if all of the genes serve a similar function, then you can all turn them on or turn them all off at the same time. It's kind of like when you have a light switch in your house that turns on multiple lights instead of just one. And so these genes can be regulated by the same on-off switch, which makes it more efficient for the bacteria. And so one example of how promoters can be turned on or off in bacteria specifically is going to be in the trip operon found in bacteria. And there is this concept of a co-repressor. So let's kind of talk about what this is. So a co-repressor is a small molecule that will bind to the repressor itself, and it will cause that repressor to bind to the operator, which is that region in between the promoter and the actual genes. So right here. And that will, again, stop or block transcription. And so here in this specific example, we're going to be talking about the trip operon. So the trip operon is a bunch of genes, so trip E, D, C, B, and A. All of these genes produce proteins that have to do with making amino acids, specifically the tryptophan amino acid. So that's why it's called the trip operon. Now, when you think about it logically, if we want to make more tryptophan, we would want these genes to be on because they make tryptophan. But if the cell has a lot of tryptophan, we don't want these genes to be on because you already have enough tryptophan. And so what's really genius about this operon is that tryptophan is the actual co-repressor. So in the presence of high levels of tryptophan, when you don't need to make any more, the tryptophan itself binds to the repressor, so it's acting as the co-repressor. And together, they're going to bind to the operator, which blocks RNA polymerase. And so you're not making these genes, which means that you're not making any more tryptophan because guess what? You have enough. So it makes sense. It's kind of like its own self-regulating operon. So if you need tryptophan, the genes are on because the tryptophan is not present to be a co-repressor. But when the tryptophan is present, then it acts as a repressor and turns off the whole system. So that is a trip operon, and it uses a co-repressor. And then the other example that's found in bacteria are inducers. Specifically, this example is the lac operon. So inducers are small molecules, kind of like co-repressors, but instead what they do is they bind to the repressor and they actually force the repressor to come off. So they're basically the opposite of a co-repressor. They bind and the repressor comes off of the operon operator. And so then transcription is turned on this time. And so the example in the lac operon is these genes. So LAC-Z, LAC-Y, and LAC-A are all genes that have to do with breaking down lactose for energy. So when a bacteria is growing in the presence of lactose, they can break down that lactose and then they can use that sugar for energy. Now, when you think about it, you only want these genes to be on if lactose is present, because these genes have to do with breaking down lactose. Now, if there's no lactose, why would you want these genes on, right? And so that's exactly how this operon works. So in the absence of lactose, the repressor is repressing. So it's bound to the operator, and so these genes are turned off. But when lactose is present, it acts as your inducer. It binds to the repressor, pulls the repressor off. And so now the genes are turned on. Because again, you want these genes on when lactose is present because their whole job is to break down that lactose. So yeah, these are just a few examples of how genes are regulated, specifically in bacteria. Obviously, in eukaryotic cells, there's other ways to do it as well. But these are just the classic ways that genes are regulated. I hope this video was helpful, and I'll see you in the next video.